In this study, we use bat MHC class I as a model to summarize the methodology and to evaluate the feasibility of a hybrid MHC class I complexed with heterologous beta-2 microglobulin.
Previous studies have shown that the mammalian B2M substitution does not significantly affect the peptide presentation, and that the hybrid MHC class I can exhibit a similar structure and function to the original molecule.
These techniques can be used to facilitate the functional and structural study of MHC I and for tissue response evaluation during infectious disease and tumor immunotherapy.
For E. coli culture transformation, add 10 nanograms of plasmid containing bat or human MHC class I beta-2 microglobulin hydrogen chain to 100 microliters of E. coli suspension and bathe the suspension in ice for 30 minutes. At the end of the incubation, heat shock the bacteria for 90 seconds at 42 degrees Celsius and return the culture to the ice bath for 2 minutes. Next, add 800 microliters of lysogeny broth to the culture and shake the suspension on a rocking platform for 20 minutes at 37 degrees Celsius and 200 rotations per minute. At the end of the incubation, spread 100 microliters of the bacteria onto an appropriate antibiotic-resistant culture plate. The next morning, pick a single recently transformed bacterial clone and inoculate the clone with 3 milliliters of LB with antibiotic medium. Place the